Hi, everyone. Thank you for joining us today. We are the Association of Cannabinoid Specialists, and we're presenting the cannabis, the gut-brain connection, and the role of the microbiome. We're very excited to have our speakers here tonight, as well as our host. I'm just going to go over a little bit about the Association of Cannabinoid Specialists. We are an international organization of healthcare professionals and industry professionals on the forefront of cannabinoid medicine. We work to advocate for the highest standards in the practice of cannabis medicine and safeguard patient care. And we're an advocacy organization striving to provide guidance to lawmakers and regulators at every level of cannabis control. We do offer professional memberships. I'll put the links in the chat for everything I talk about, but you can visit that link to sign up for just a donate what you can contribution. Our member benefits include a $85 annual subscription to cannabis and cannabinoid research, which is usually $544, access to a range of handbooks, tutorials, and a clinical reference library that's curated by our doctors, 20% discounted pricing to ACS courses, networking events, and a lot more. So if you're interested in signing up, I'll put my email as well as the link in the chat for you. We do rely on donations from supporters as a nonprofit organization. Donations are tax deductible, so please, if you'd like to, I'll put the link in the chat for that as well. And it's also right here. It is Giving Tuesday, so just consider that when making your donation. We really appreciate everything that you guys do throughout the year, so thank you. And I'm going to turn the floor over to Dr. Abodeely, and he will introduce our speakers.
All right, good evening, everyone, and welcome to the ACS Educational Symposium. My name is Dr. Adam Abodeely, and I'll be the moderator as well as one of the presenters here this evening for the symposium, which is titled Cannabis, the Gut-Brain Connection, and the Role of the Microbiome in Cannabinoid Medicine. And, you know, we've come a long way in understanding the role of the human microbiome, while at the same time we're understanding a lot more about cannabis and cannabis physiology and pharmacology. And there definitely appears to be an intersection between the human microbiome and the endocannabinoid system and the gut-brain connection and the function and physiology and health and wellness of the GI system. So this presentation tonight, I hope will be able to expand on some of the the future aspects of cannabinoid medicine, but also kind of where we are at currently. I'm very thrilled to have our guest lecturers here tonight, Dr. Keith Sharkey, Dr. Mahesh Mohan, who unfortunately, due to a family emergency, could not make the presentation. However, he was generous enough to record his presentation, and so I will share his presentation during his speaking slot. And then Dr. Michael Camilleri as well. We are very fortunate to have all of these researchers on this panel tonight, as these really are several of the leaders in this field and emerging concepts of this field of the human microbiome and the role that the endocannabinoid system plays in our overall health and wellness. So our presentation tonight will be 4 separate talks, both around 15 to 20 minutes each. I will introduce each one of the lecturers. Sorry here. I will introduce each one of the lecturers before they speak. I will be briefly chatting about the history of cannabis in gastrointestinal health and wellness. We'll briefly review the endocannabinoid system as it relates to GI anatomy and physiology, and what happens in dysfunction of the endocannabinoid system in particular GI diseases. We'll explore the current role of cannabinoid medicine for gastrointestinal disease, and then we're going to be hearing from some of the other lecturers that I hope are going to get us thinking about some of the aspects of the future of cannabis in GI care and as it relates to the role of the human microbiome. Briefly about me, again, my name's Dr. Adam Abodeely. I'm a colorectal surgeon in upstate New York and Lake Placid, New York. I specialize in gastrointestinal cancers, inflammatory bowel disease, and motility disorders. And I've been in practice for 15 years now, but I've been working with medical cannabis patients since 2012. I also work closely with the New York State Office of Cannabis Management on their Medical Cannabis Advisory Council, helping provide guidance for cannabis providers in the state of New York as it relates specifically to GI conditions. When we look at the history of cannabis for medical conditions, GI conditions are some of the most early reported indications for cannabis use. We look back in, several thousands of years ago when the ancient Egyptians were described using cannabis for things like hemorrhoids, and they used suppository fashions, preparations of these at the time. And throughout history, we've seen cannabis be used for GI conditions. But what I find interesting is that it wasn't until over the past 100 years that we had a name for a lot of these, even a lot of these diseases. So when we look through history, we see a lot of documentation of cannabis being used for often what was described as pain of the innards. Well, pain of the innards, we know, without modern day diagnostic imaging and workups, we don't know what they were actually referring to as pain of the innards. It most likely was a lot of the same diseases and conditions that we're using cannabis for today, such as inflammatory bowel disease, but they probably were also using it for things like diverticulitis and irritable bowel syndrome or just unexplained abdominal pain. But either way, cannabis has played a role in GI health, throughout history. And the endocannabinoid system plays a very important role in our GI physiology. And we know that the job of the GI system is to digest our food, but it also detoxifies our system. It eliminates waste, and it's extremely important in immunity and metabolism as well. And we're learning how the endocannabinoid system can really be manipulated to affect these different physiological processes. And although we talk about, I show this slide just to show the several layers of the intestinal tract, but when we think about the GI system, we have to think about things other than just the intestines themselves. So they'll largely, what we refer to in the studies we're looking at are largely based upon the intestinal aspect of it. But just keeping in mind that the GI system also consists of the pancreas, the liver, and and helps control metabolism as part of the overarching theme of the GI system. But even when we look at just the layers of the intestinal tract, we see a variety of different anatomical locations, and components, everything from veins, arteries, nerves, muscles, our epithelium. And what we have identified is that all aspects, every component of the endocannabinoid system, whether it be the naturally occurring endocannabinoids themselves, the receptors which bind these endocannabinoids, and along with the metabolic enzymes which either produce the endocannabinoids or degrade them, have all been demonstrated and found to perform functions within all aspects of these anatomical locations. And what we know, broadly speaking, when we speak broadly about cannabinoid interactions, we know that it tends to have a largely inhibitory effect on the GI tract at large. We know that CB1 activation tends to decrease motility. Activation of CB2 receptors doesn't tend to directly affect motility, but in states of inflammation, activation of CB2 receptor can also assist in decreasing motility, which is induced by the inflammation. We also know that activation of the cannabinoid receptors can decrease intestinal secretions, decrease intestinal inflammation, decrease pain, and even decrease acid secretion. So when we think about GI diseases in the context of the symptoms and the variety of different outcomes, it wouldn't be abnormal to think that dampening some of these activities, such as motility and secretion and inflammation, would have a bad effect. I mean, we know that anytime we can decrease motility, decrease pain, decrease inflammation, decrease secretions, we have the ability to potentially affect the symptomatology or even the disease process of a variety of different GI conditions. And this is one of my favorite tables, just because it really shows the fact that when we look at a disrupted or when we look at disease, and every disease of the GI system, whether it be things like diverticulitis, celiac disease, inflammatory bowel disease, and cancers, we've identified disruptions in the endocannabinoid system. In some cases, these disruptions or these alterations are caused by the disease, but we also know that in some cases there's genetic polymorphisms, particularly of the cannabinoid 1 receptor, for example, which might be partially responsible for actually driving the phenotypic expression of a lot of these diseases and the symptoms associated with it. We've seen this particularly with irritable bowel, where we've seen a defect of genetic polymorphism of the CB1 receptor, for example. So in my practice, and really in most GI practices that may utilize cannabis, we use cannabis for a variety of different things, and I'm just going to go through some of these really briefly. Cannabis for cancer is where I began my journey with my patients, and we have very good evidence base that symptom management can be beneficial for cancer patients, whether it's as a result of their chemotherapy-induced nausea and vomiting, whether it's to help stimulate appetite or maintain weight. The pains that can be associated with either the cancers themselves or the chemotherapies, which can cause neuropathies. And we've also seen cannabis provide just an overall improvement in quality of life and other aspects of quality of life, such as general mood and sleep. So cannabis for cancer care is, even in the GI world, is really supported as a palliative and an adjunctive option. Is cannabis chemotherapy? Well, we certainly know that cannabis has several different anti-cancer properties. And although while the cannabinoids do exhibit several of these anti-proliferative and anti-carcinogenic effects, we still need further research to really determine the role of cannabinoids as a true cancer treatment. So for right now, in the world of cancer, that's largely used as an adjunctive therapy for symptoms. The next large category in GI is inflammatory bowel disease, and as mentioned, I've been working with New York State on this, and we're just about to publish a large review on the use of medical cannabis for inflammatory bowel disease. And what we've learned from this review, it's been similar to several other reviews on this topic, but what we know is that symptom relief with cannabis is quite consistent and can be very useful for improvement of symptoms of abdominal pain, irritability, appetite, sleep, and general quality of life. What we haven't been able to prove yet is that there's any evidence for remission of the disease or mucosal healing with the use of cannabis directly. We also know that formulation and dosing matters, and there are also several different challenges which GI patients might face when using cannabis for inflammatory bowel disease. We also know that we need some larger trials to really still figure this out, but cannabis is considered to be an effective adjunct for symptom management in inflammatory bowel disease patients, particularly those who have failed other alternative therapies. And we've seen that similar in our practice. We did an observational study from 2018 to 2020 where we looked at 42 patients. 35 we were able to study out to 12 months, and we saw similar improvement in things like Crohn's disease activity index and improvements in quality of life. As I mentioned, the endocannabinoid system, plays an important role in the physiology and motility of our GI tract. So it shouldn't be any surprise that people with irritable bowel syndrome who can suffer from things like hypermotility, increased abdominal pain, increased diarrhea might benefit from cannabis. And again, we've shown that to be particularly the case in particular patients who have genetic variants in the endocannabinoid receptor, the CB1 receptor, as well as the Fa enzyme, which is responsible for breaking down our endocannabinoids. And certainly what we've seen is, even looking at clinical trials using dronabinol, we've showed that it's been shown to have reduced fasting colonic motility and increased colonic compliance, and these can all are what are thought to be responsible for the general improvement in symptoms with activation of our cannabinoid receptors. Dr. Camilleri will be talking about gastroparesis further, but our practice is also a referral center for patients with refractory gastroparesis. And despite the fact that we know that activation of the cannabinoid receptors can slow motility, we find that it's quite frequently beneficial for patients, again, particularly when we're trying to control symptoms in light of other failed modalities. But Dr. Camilleri will share some more information on that in a moment. We've also seen the use of cannabis for cyclical vomiting syndrome. Again, it's a complex condition, but it's particularly because the etiology is multifactorial. But there are several cases where cannabis might provide relief. However, we also know that could be a double-edged sword in the sense that some people can develop cannabinoid hyperemesis syndrome, which sometimes there's some overlapping symptoms with cannabinoid hyperemesis syndrome and cyclical vomiting syndrome, but it still in select patients can be beneficial. And in addition to abdominal pain, I've also found it to be very effective for patients with a variety of different forms of pelvic pain, including proctalgia and tenesmus. And that can be a result of either its ability to affect motility, rectal compliance, or control inflammation locally in the cases of inflammatory states. So we know that cannabis does offer meaningful benefits for patients in the GI world. But despite this, the clinical responses for any of us who've worked with cannabis know that the outcomes can be variable as it relates to efficacy, as it relates to side effects. Dosing, we know, is not universal yet. We know that route of administration matters. Some people tolerate inhalational, where they don't tolerate oral preparations, and vice versa. And of course, we know that those chronic cannabis users or those with elevating or escalating doses can develop tolerance as well. So we have a ways to go when it comes to determining standardized dosing and formulas. But also, I think there's going to be some opportunities down the road for product development in ways that we can improve upon the poor and bioavailability of the majority of the oral cannabinoids. It'll be interesting. A lot of the side effects of cannabis are centrally acting. So if we can, once we understand the activities peripherally, then we can start to look at some peripherally restricted cannabinoids, which won't have a centrally acting effect, but might still affect the gut. And we're moving into an era of personalized care. And we know right now you can have your own human endocannabinoid system tested to see if you have enzyme genetic polymorphisms, or if you're a slow metabolizer or fast metabolizer. But at the same time, we're also we're entering a world where the gut microbiome is beginning to be looked at. And I think that there's a great deal of data out there that show that intestinal microbiome can truly influence things like cannabinoid metabolism and inflammatory tone and this gut-brain signaling. So in order for us to better understand, the interaction, the interactions between cannabis and our GI system, I think we're going to have to start looking at the gut and the microbiome as well. And hopefully we will be able to find ways to enhance efficacy, reduce side effects, and refine dosing strategies in a way that are more predictable and personalized for patients. When we look at cannabis in the human microbiome, the effects it has in the human microbiome, what we do know is that cannabis can change the human microbiome. What we don't yet know is what are the true implications of that and whether or not there are ways that we can capitalize on that for the sake of cannabis care. So there's this complex interaction going on between, the endocannabinoid system, our brain, the gut-brain connection, our intestines, cannabis, the intestinal microbiome. So with that said, I'm going to introduce Dr. Keith Sharkey here. Dr. Sharkey, do you want to share your screen with us? Dr. Sharkey will be sharing more about the microbiome and the gut-brain connection. Dr. Keith Sharkey was educated at the University of London and the University of Liverpool. After his postdoctoral training in Hungary, the UK, and Canada, he was appointed as an assistant professor in the Faculty of Medicine at the University of Calgary. He's remained at the University of Calgary for his academic career and is currently a professor of physiology and pharmacology at the Cummings School of Medicine at the University of Calgary. And we're excited to have Dr. Sharkey here tonight because his research is focused on understanding the physiology and the pathophysiology of the neural control of the gastrointestinal tract and the brain-gut interactions in health and disease. His current interests include the studies of the role of the enteric nervous system in GI pathophysiology, the endocannabinoid system of the gut-brain axis and control of gut functions, and the role of the GI tract in the development of ALS. He has published over 280 original peer-reviewed research articles and reviews, and for his contributions, he was elected as a Fellow of the Canadian Association of Gastroenterology, a Fellow of the Canadian Academic Health Sciences, and a Fellow of the Royal Society of Canada. So with that, Dr. Sharkey, the floor is yours.
Well, thank you very much. It's a very generous introduction. I hope I warrant it. So I'm going to talk to you a little bit about the microbiota gut-brain axis and its relation to the endocannabinoid system to try to set the stage. But actually, Adam did a terrific job already. So before I start, I should just add that I have no disclosures or no disclosures at all, actually. And my lab is generously funded through the federal funding agencies in Canada, but also philanthropic agencies that support research. So we've heard about the gut from Adam. The gut is not only an organ of digestion, but a very important organ of defense. And when we think about the gut and the brain, we have to sort of think a little bit about what the gut does and how it functions. And when you do that, you realize the gut has to store and move food along its length, and it's very long. It has to digest that food in the right place at the right time, absorb the nutrients and water, and store and eliminate waste. And when you think the spatial temporal control of digestion is very non-trivial. It's quite a complicated act requiring substantive control, and it literally takes a brain to detect the food, initiate and control motility and the processes of digestion, and make sure everything happens at the right place at the right time, or else we end up either losing nutrients or with problems of the digestive process. But at the same time, the gut has to defend itself. The enzymes, the acid, and the activities that will digest a steak or a McDonald's or whatever you choose to eat, maybe a vegetarian meal, will digest you just as well. And you've got to protect that and keep it inside the lumen of the gut. The food that we eat is not sterile. When it's broken down, it breaks down into antigens that could give rise to immune responses. And in addition to parasites and other infectious agents, the gut has to protect you from them. And as we're going to hear in a couple of minutes, it hosts an enormous rich microbiota. Adam introduced that. And if those get out of the gut and into you, they cause infection and sepsis just as well as infectious agents. And it literally takes a brain to respond rapidly to the luminal challenges to coordinate an effective series of host defense mechanisms. In fact, not one brain, but two brains. The brain in our head that we think about as the brain, but also the brain in the wall of the gut. The nerves that innervate the gut turn the myenteric and submucosal plexuses. The gut is a tube. If you cut a tube along its long axis and lay it out flat, it ends up looking something like this in a schematic way. And the neurons, which are colored red and green here, so apologies to the red-green colorblind people, but those richly innervate the entire gut, and those are functional and supported by enteric glia, which are the little yellow cells. The entire gut is richly innervated. The idea that the nerves in the gut and the nerves in your head, the nerves in the central nervous system, interact isn't new, but it was kind of formalized in the 80s, the 1980s, and culminating in this concept, which we call the gut-brain axis, illustrated from this slide in a nice review from 1990, illustrating how the activities of digestion and defense are regulated in this bi-directional way. The important thing are the double heads on those arrows. But then in the 2010, the early to mid 2000s, really culminating in 2010, as a result of the sequencing efforts of the human microbiome, we realized that the gut is an incredibly rich source of microbes. We knew that the gut contained microbes before that time, but now we know it's an incredibly rich and diverse source. And in fact, the human microbiome is not restricted to the gut, but the gut has the largest proportion of microbiomes at any site in the body. Its metabolic output is so vast, actually, that it essentially becomes an organ in itself. So the gut-brain axis is now the microbiota gut-brain axis. The bugs in the gut start in the stomach. The stomach is not sterile as it was originally thought, but the quantity of quantity of bacteria in particular increase dramatically as you go from the stomach down to the colon. These bugs make enormous quantities of metabolic products that are biologically active and can travel through the bloodstream or by activation of the nerves of the gut to impact the brain. And we can see here in a schematic of the wall of the gut illustrating the relationship between the gut microbiota and the signaling entities of the gut, which are the enteroendocrine cells that line the wall of the gut, that talk to the enteric nerves and the extrinsic nerves, that molecules released in the lumen of the gut or indirectly via enteroendocrine cells or immune cells can play a critical role in the regulation of gut function. And you can think about this, and Emma and Maya has done a nice job of illustrating how, again, in a bidirectional sense, microbe-derived molecules and gut-derived molecules talk to one another to regulate the functions of the gut, as well as the functions of the brain. And the whole thing is this rather interesting, multi-directional network. Of course, the bugs in your gut are modulated by a variety of things, including elements released from the wall of the gut, such as antimicrobial peptides, which are released from Paneth cells, for example, in the small bowel. But diet is by far the most important modulator of the gut microbiota. And we can see here in this recent review from John Cryan's group that proteins, dietary ingredients like polyphenols, prebiotics, and dietary fibers have a positive effect on the gut commensal bacteria to regulate the brain-gut axis, whereas Western diet typically has a pro-inflammatory and rather negative effect on the gut bacteria, and these altered bacteria may contribute in whole or in part to the rather negative effects, the pro-inflammatory effects of Western diets. A few terms that may have come up in your reading or that you may be interested in understanding. Prebiotics are the food of bacteria, if you will. In the diet, these things such as fibers can sustain long-term changes in the composition of the gut bacteria. Another term is probiotics. Probiotics are live bacteria that you consume. You may see them in the grocery stores, in yogurts or in other foods, or as supplements. And they're usually short-lived and transient when they don't usually colonize the gut. So if you take them for a day, they're gone the next day. If you continually take them, they can have biological actions. A symbiotic is a combination of pre and a probiotic. And there's something called postbiotics. Those are the compounds produced by bacteria. They may be supplements and that you may see in the various health food stores. We already heard nicely from Adam about the cannabinoid system, and this brain-gut axis, the endocannabinoid system plays a very important modulatory role, and it impacts all aspects both centrally and peripherally. Since he did such a great job, I'm not going to talk about that, but I am going to answer the question, at least I hope, in a couple of minutes, is there a link between the gut microbiota and the endocannabinoid system? And I think the answer is yes. Direct evidence of this started to appear quite early on when, for example, this group in Belgium demonstrated that if you perturb the gut microbiota, whether you do so in obesity and you feed animals prebiotics, this is animal studies, whether you give antibiotics to eradicate gut bacteria, or in the germ-free state and then you reintroduce gut bacteria, you alter the expression of the endocannabinoid receptors. This is all normalized data. That's why it can all go in the same direction. But you do so in a region-specific manner. That's an important thing. In this case, the colon was affected, whereas the jejunum wasn't. And this is just one example, but there's lots of them in the literature, or there's some at least. If you alter the cannabinoid receptors, so this is a bidirectional thing, so now you block the CB1 receptor, you can see that blocking the CB1 receptor in the context of a high-fat diet can alter the population of bacteria in the gut. And these are three examples, and they go in different directions. Akkermansia goes up, whereas Lactobacillus and this bacteria that I cannot pronounce easily this evening goes down. So this is it's a very complex interrelationship, but the endocannabinoid system can regulate our bacteria and vice versa. What about exogenous cannabinoids? Because those are important, as Adam has indicated, as medicines. And one of the things that's rather interesting in an epidemiological sense is that cannabis users tend to be lean. This has been borne out by multiple studies, including very, very rigorous studies using NHANES data, for example, in the United States. And the question is, why is that? Because if you consume a cannabis acutely, many people develop the munchies, or a desire to increase food intake. So you'd think maybe cannabis users wouldn't be lean, but the opposite. But that's not true. They're lean across. This is, of course, on a population level. So we asked the question, what about cannabis use and gut bugs? And a few years ago now, and in lean animals, when we give THC, the active ingredient of cannabis, as you know, doesn't do a lot to the bacterial content. But interestingly, when we gave animals a high-fat diet to make them obese, the THC markedly altered their ratio of the major families of bacteria in the gut. And just as we saw before, this bacteria, Akkermansia, is one that's quite important in homeostasis in the body. It elevated this quite significantly. And whether it was a human taking cannabis, or in this case, mice, we actually reduced their body weight. So even when they were taking a high-fat, high-sugar diet, they actually lost weight and became lean. THC is not the only cannabinoid in cannabis. Another important one is cannabidiol, which you can take as individually or it's part of the cannabis medicine. A recent study just published in February illustrates that cannabidiol selectively increases this particular bacteroidetes species. Now, this is in a mouse, but we have no reason to think that similar things wouldn't occur in a human. And in this case, the study was designed to look at muscle types within the animal and demonstrated that actually animals given CBD had greater endurance to exercise as a result of biochemical changes to the muscle composition. And these were mediated by branch chain amino acids, specifically produced as a metabolic product of the bacteria that was enhanced. And actually, I saw that Adam showed this slide too. A number of studies in humans now have demonstrated effects of cannabis, which they call marijuana, which is a pejorative term. It's much better to use the word cannabis, as well as endocannabinoids can alter in a variety of conditions. So the gut microbiome regulates the endocannabinoid system. The gut microbiome and the endocannabinoid system are concomitantly regulated by diet. Cannabinoids can alter the gut microbiome. And so to conclude my little presentation, I would say that the gut microbiota gut-brain axis is the essential framework to understand an individualized response to cannabinoid therapies. There's an important bidirectional link between the gut microbiota and the endocannabinoid system that impacts whole body physiology in both health and disease. And understanding these relationships will allow physicians, surgeons like Adam, physicians like Dr. Camilleri there, who'll speak in a few minutes, to really understand how these can be used as medicines, or when individuals take them either medicinally or recreationally, can impact their physiology and their behaviors as well. So I will end there. and thank you very much for listening.
Fantastic. Thank you, Dr. Sharkey. Our next, we're going to save all the questions I'd ask. If you have any questions for any of the presenters tonight, please leave them in the chat box, and we will try to answer the questions at the conclusion of the final presentation. Our next presenter is Dr. Mahesh Mohan, and as I may have previously mentioned, unfortunately, due to family emergency, he could not make it and had to travel, but was kind enough to send his presentation via a recording here. So before I start his presentation, I will just introduce him. Dr. Mahesh Mohan is a professor at the Texas Biomedical Research Institute and a core scientist at the Southwest National Primate Research Center in San Antonio, Texas. He leads the Disease Intervention and Prevention Program at Texas Biomed and Infectious Diseases Immunology and Control Unit of the SNPRC, which is the Southwest National Primate Research Center. He also co-directs the Basic Science Core of the Texas Center for AIDS Research. Dr. Mohan holds a Bachelor of Veterinary Sciences degree from Nadu Veterinary and Animal Sciences University in India, and an MS in Reproductive Biology from the University of British Columbia, and a PhD in Reproductive Biotechnology from Oklahoma State University. In September 2003, he joined the Tulane National Primate Research Center as a postdoctoral fellow to work on HIV pathogenesis using the non-human primate model of HIV infection. He advanced through the academic ranks, becoming an assistant professor in 2010 and an associate professor in 2016. In 2019, he moved to the Texas Biomedical Research Institute as a full professor. His ongoing research focuses on epigenetic mechanisms, specifically non-coding RNAs and DNA methylation that contribute to HIV or SIV-induced gastrointestinal and neurologic dysfunction. His current work also explores combined therapies using phytocannabinoids and antiretrovirals to reduce inflammation and related comorbidities, including a newly NIH-funded study to investigate the use of focused ultrasound-facilitated blood-brain barrier opening for the delivery of long-acting ART drugs and gene therapy vectors to the brain. So with that said, I will play Dr. Mohan's presentation now.
Good evening, everybody. Before I begin, I would like to thank the organizers for the invitation to present our findings on the use of brain access.
Okay, just one moment.
In HIV and SIV infection at the ACS symposium. So with the advent of modern antiretroviral therapy, people with HIV are living longer, but they are at a higher risk of developing inflammation-driven comorbidities like HIV-associated neurocognitive disorders, HIV-associated cardiovascular disease, etc. The persistent intestinal dysfunction, along with antiretroviral therapy toxicity, loss of regulatory T cells, and the activation of opportunistic infections like cytomegalovirus can maintain
Adam, I apologize. I think you have to turn on your mic.
strong need to explore alternatives for use as adjunct therapies to ART drugs to reduce ART toxicity and chronic inflammation. We're currently studying HIV-associated neurocognitive disorders, also known as HAND. What you see common in HAND, Alzheimer's disease, Parkinson's disease, and multiple sclerosis is neuroinflammation arising from persistent activation of microglia, leading to neuronal loss and eventually cognitive decline. A major driver of neuroinflammation is persistent type 1 interferon responses that is common to all four diseases. What you see here is the cross-section of a blood vessel in the brain that comprises the blood-brain barrier that is formed by the tight junctions between adjacent endothelial cells together with astrocytic food processes and pericytes that blocks large molecules like anti-cancer and some antiretroviral therapeutic drugs, including therapeutic antibodies from crossing the blood-brain barrier. Interestingly, SIV or simian immunodeficiency virus infected rhesus macaques administered chronic low-dose delta-9 tetrahydrocannabinol or THC survived longer than their vehicle-treated counterparts, even in the absence of antiretroviral therapy, suggesting that it is chronic immune activation or inflammation and not viral replication that drives disease progression. So since cannabinoids are highly lipophilic, we hypothesized that it would easily cross the blood-brain barrier and attenuate pro-inflammatory type 1 interferon responses in the brain. So we tested our hypothesis in the SIV-infected rhesus macaque model of AIDS without and with antiretroviral therapy. In these studies, we initiate THC at 0.18 mg per kg. 4 weeks later, on the day of SIV infection, we increased the dose to 0.32 mg per kg that was maintained for the rest of the study. Antiretroviral therapy, consisting of these three drugs, emtricitabine, tenofovir, and dolutegravir, administered subcutaneously, was initiated at 2 weeks post-SIV infection and continued until 6 months post-infection. We collected blood, cerebrospinal fluid, intestinal resections, in this case, jejunum resections, and again, all of these samples, including brain at necropsy. Now, the jejunum resections were used for gene expression and microbiome studies. Microbiome profiling was performed using shotgun metagenomic sequencing. These two heat maps here show type 1 interferon-stimulated gene expression in the brains of uninfected controls on the left, SIV-infected rhesus macaques treated with vehicle on the right, or THC in the middle, marked with this black box. And all of these animals did not receive antiretroviral therapy. Interestingly, the 5 animals that received THC have fewer red squares for the 26 statistically significant type 1 interferon-stimulated genes compared to the 6 vehicle SIV rhesus macaques that have markedly a higher number of red squares here, showing that these type 1 interferon-stimulated genes are significantly upregulated. In addition, we detected high levels of 2 neuroprotective metabolites, glycerophosphoinositol and glycerophosphorylcholine, in the plasma of THC SIV rhesus macaques relative to the uninfected controls. The take-home message is that low-dose THC can cross the blood-brain barrier and successfully reduce type 1 interferon responses in the brain. Additionally, cannabinoids can exert beneficial effects in the brain via its ability to modulate tryptophan metabolism. Now, tryptophan is an essential amino acid that is not synthesized by your body, and so it must be provided in the diet. Approximately 90% of the absorbed tryptophan from the intestine enters the kynurenine pathway that is catalyzed by this enzyme called IDO1, which stands for indolamine 2,3 dioxygenase. About 7% of the absorbed tryptophan is converted to indoles. That includes the neuro and cardioprotective indole 3-propionate, and the remaining 3% is converted to serotonin. Now, we previously showed that long-term low-dose THC can inhibit the activity of IDO1, thereby making more tryptophan available for conversion to serotonin and other indole metabolites like indole 3-propionate. We tested this hypothesis in a separate cohort of ART-naive and ART-treated SIV-infected rhesus macaques receiving either vehicle or THC. First, we looked at the plasma metabolome profiles. This PCA plot shows that the global metabolic signature of the vehicle ART rhesus macaques in green symbols showed better separation from their respective pre-infection samples shown in light gray symbols. In contrast, the THC ART group shown here in pink symbols demonstrated the least separation from their respective pre-infection samples shown here in dark gray symbols. The plasma viral loads were undetected around 2 months post-infection, suggesting very effective viral suppression by our triple drug ART cocktail. The table below shows the total number of metabolites detected in the vehicle and the THC ART groups relative to their respective pre-infection samples, and then between the THC and the vehicle group. So the circles in both of these metabolic pathway maps represent 20 tryptophan metabolites detected in the plasma of vehicle ART and THC ART rhesus macaques at 5 months post-infection compared to their respective pre-infection samples. Interestingly, IPA levels, as shown in pink circles, did not significantly change in the vehicle ART group. But THC added as an adjunct to ART significantly increased IPA levels as revealed by the dark red circles. And it's also very clear in this box plot shown here. Now, even though kynurenine levels were significantly higher in the THC ART group, kynurenine and tryptophan ratios, which is a better indicator of IDO1 functional activity, did not show significant changes between the vehicle and THC ART group at 5 months post-infection. Similar to IPA, THC significantly increased serotonin levels. Now, since pre-infection serotonin levels were low in the THC ART group, we combined both pre-infection samples and found plasma serotonin levels to be significantly only in the THC ART group compared to the pre-infection samples from both vehicle and THC group. Next, we dissected the serotonin signaling pathway in detail and detected a higher number of green staining serotonin-producing enterochromaffin cells in the jejunum of THC ART compared to the vehicle ART rhesus macaques at 5 months post-infection. The image below shows a close-up view of the serotonin-producing enterochromaffin cells, and image quantification detected significantly higher number of serotonin-positive enterochromaffin cells in the jejunum of THC SIV ART rhesus macaques compared to the vehicle ART and in both pre-infection time points. Now, serotonin acts via binding to serotonin receptors. After mining omics data from one of our previously or recently published studies in JCI Insight, we found serotonin receptor 4, also known as 5-HTR4 and 5-HTR7, which is the serotonin receptor 7, out of the 7 known receptors to be significantly upregulated in the jejunum of THC SIV rhesus macaques. Now, serotonin acting via HTR4 has been shown to reduce inflammation in mice with colitis. Using immunofluorescence staining, we found HTR4 protein expression to be significantly increased in the jejunum of vehicle ART rhesus macaques compared to their respective pre-infection time point. However, the THC supplemented group showed even higher HTR4 expression compared to their pre-infection time point and the vehicle ART rhesus macaques, which is very evident from this quantification plot. So this is a clinically relevant finding because HTR4 agonists are considered as potential treatment options for patients with inflammatory bowel disease and irritable bowel syndrome. The protein expression of HTR7 was also significantly increased in the jejunum of THC, of actually vehicle ART rhesus macaques at 5 months post-infection compared to their pre-infection time point. Now, similar to HTR4, combining ART with THC further increased HTR7 receptor protein expression. As you can see from this quantification plot, THC supplementation significantly increased HTR7 protein expression compared to both vehicle ART and both pre-infection time points. Like HTR4, HTR7 has also shown anti-inflammatory effects in the intestine. Next, using in vitro cultured human small intestinal epithelial cells, we found that THC dose dependently increased both HTR4 and HTR7 protein expression, shown here in green, with the highest expression detected with 3 micromolar THC for both HTR4 and HTR7. Next, using specific antagonists of the cannabinoid receptor 1, AM251, and cannabinoid receptor 2, AM630, we found that THC-induced HTR4 and HTR7 protein expression, mainly acting via the cannabinoid receptor 2, as blocking cannabinoid receptor 2 with AM630 reduced protein expression of both HTR4 and HTR7, as shown here in this quantification plot. Next, we investigated HTR4 and HTR7 protein expression levels in the ganglion cells of the myenteric or box plexus located between the inner circular and the outer longitudinal muscle layers of the intestine, where the vagus nerve innervates, as this is a key piece of the microbiota gut-brain axis communication. The immunofluorescence staining confirmed strong HTR4 and HTR7 protein expression in the myenteric plexus neurons. The red represents neuronal nuclei, which is a marker of neurons. And this confirms that the staining was detected only in the ganglion cells. The blue is a marker for nuclei, which is DAPI. The serotonin produced by the enteric enterochromaffin cells can act on these receptors and signal to the brain via the vagus nerve. So this represents physical signaling along the or via the vagus nerve along the gut-brain axis. And THC can enhance the signaling, basically physical signaling through the vagus nerve along the gut-brain axis. Using shotgun metagenomics sequencing, we found significantly high relative abundance of Lactobacillus plantarum, which has previously been shown to stimulate serotonin production in the intestine. So these two papers here, the first one shows the importance of the serotonin HTR4 receptors in maintaining cognitive function. And the second paper published in Cell Reports describes the role of intestinal bacteria in serotonin production. Interestingly, our findings suggest that both can be achieved using low-dose phytocannabinoids. In the intestine, IPA or indole 3-propionate has been shown to strengthen the epithelial barrier and reduce inflammation, and its plasma levels are associated with higher gut microbiome diversity. Increased IPA levels can also reduce neuroinflammation and potentially slow down cognitive decline, not only in HIV, but also other neurodegenerative diseases like Alzheimer's disease, where it has been prescribed as a supplement called Oxygon. Unlike serotonin, IPA is only produced by the bacteria present in the colon. As evident here, THC significantly increased plasma IPA levels even in the absence of antiretroviral therapy. So out of the 7 known bacterial species that carry the enzymatic machinery to synthesize IPA from tryptophan, the relative abundance of 3 species, namely Clostridium botulinum, Clostridium paraputrophycum, and Clostridium cadaverus, were significantly high or showed significantly high relative abundance in the colonic contents of THC Ascivariosis macaques compared to uninfected controls. Only one species, Clostridium sporogenus, was found to show high relative abundance in the colonic contents of vehicle Ascivariosis macaques compared to uninfected controls. So here, by inhibiting inflammation, THC can maintain anaerobic conditions that allows obligate anaerobic commensals like Clostridia to flourish in the colon, more specifically in the descending colon, and produce these beneficial metabolites like IPA that can enhance gut-brain axis signaling. And these findings were published in this paper shown here. Next, to further explore IPA signaling mechanisms, we investigated the expression of the IPA receptor, aryl hydrocarbon receptor, also known as AHR, and found its protein expression to be significantly increased in the jejunum of THC, SIV, ART, rhesus macaques, compared to both pre-infection controls and the vehicle ART rhesus macaques, which is again shown in this quantification plot below. Like serotonin receptors, THC dose-dependently increased AHR protein expression in vitro cultured human small intestinal epithelial cells with 3 micromolar THC inducing the maximum AHR protein expression, which is again evident here in the quantification plot. To confirm the completeness of the microbiota gut-brain axis signaling, we stained and detected significantly increased AHR protein expression in basal ganglia neurons of THC ART rhesus macaques, shown on the bottom row compared to uninfected controls shown in the top row, and vehicle ART rhesus macaques shown in the middle row. So this again represents chemical signaling, wherein IPA released from the gut can enter the bloodstream and then reach the brain, cross the blood-brain barrier, and exert protective effects pretty much in the brain, more specifically inhibiting neuroinflammation in the brain. The data presented in this talk have been published and can be found in the 4 publications shown here. In summary, low-dose cannabinoids can positively modulate the microbiota gut-brain axis by inhibiting neuro and intestinal inflammation, including intestinal dysbiosis. By inhibiting IDO1, THC can make more tryptophan available for conversion to serotonin and IPA, which can signal to the brain via the vagus nerve for serotonin and indirectly via the bloodstream for IPA to suppress neuroinflammation. I'm also happy to say that some of our NHP studies have resulted in clinical trials led by Dr. Ali Janabian, who is a good friend of mine at McGill University, as shown here, and their encouraging findings have prompted them to plan for larger clinical trials in the future. So finally, I'd like to acknowledge my lab members at the Tunel National Primate Research Center, where I worked from 2003 to 2019, and then my current lab members at the Southwest National Primate Research Center in San Antonio, Texas. Bulk of the work shown here was performed by Dr. Lakshmini Premadasa. And I'd also like to acknowledge the veterinary medicine staff at the TNPRC and the SNPRC, my collaborators at the LSU Health Science Center, New York Medical College, University of Nebraska, and finally the NIH for the much needed funding in the form of these R01 grants, without which these studies would not have been possible. I'd like to stop here and answer any questions if you have. Thank you very much.
All right, Dr. Mohan, thank you very much for that presentation. Some very interesting research. Our final presenter of the evening will be Dr. Michael Camilleri. Dr. Camilleri, you can share your screen whenever you can, and I will introduce you here. Dr. Camilleri is professor of medicine, pharmacology, and physiology, and consultant in the Division of Gastroenterology and Hepatology at the Mayo Clinic in Rochester, Minnesota. He received his medical degree from the University of Malta Medical School and his DSc degree from Imperial College, London, England. His research interests include clinical enteric neurosciences, gut neurohormonal control, obesity, irritable bowel syndrome, and pharmacology and pharmacogenetics. He has published approximately 1,300 articles, and he has mentored more than 80 national and international postdoctoral fellows and scientists. He is the past president of the American Neurogastroenterology and Motility Society and the American Gastroenterological Association. And he is currently an associate editor of the journal Gut. So with that said, Dr. Camilleri, the floor is yours.
Thank you. A lot of what I was planning to say has already been beautifully covered by the other speakers, but the objectives I have are to address questions of what are the endocannabinoids, what are their systems of synthesis and degradation, where are the receptors localized, how do they work, perhaps to improve GI functions, and then potentially to examine experimental studies looking at human cannabinoid therapy, particularly in disorders of motility, pain, and functional disorders, and finish off with a trial that we performed with the help of an NIH grant to look at the effect of cannabidiol in patients with gastroparesis. So let's start. What are the endocannabinoids? And much of this, of course, is well known to all of you who have interest in cannabinoids. So the endocannabinoids, such as AEA and others, are synthesized to acylglycerol being the other main one, are synthesized in the postsynaptic neuron, and then there are enzymes that are involved in their synthesis, like NAPLD and diacylglycerol lipase A. And so from there, the endocannabinoids traverse through the synaptic cleft into the presynaptic membrane, and therefore they are retrograde messengers binding to presynaptic receptors such as CB1 and CB2, GPR55, and very importantly, the STRIPV1 receptor, which is very much involved in pain sensation. But once they are bound to these receptors, through various effectors, they reduce the activity of protein kinases, and they modulate channels as well as monoamine neurotransmitter release. And of course, for those of us who are interested in motor function in the GI tract, acetylcholine, a excitatory transmitter, and norepinephrine, generally an inhibitory transmitter, are very, very important. So after having had their effect on the presynaptic membrane and tissues, the endocannabinoids are then reuptaken by the postsynaptic neurons and hydrolyzed by fatty acid amide hydrolase and monoacylglycerol lipase. Now, it's important to remember that one of the actions that we're going to see later from the effects of cannabidiol in particular is to inhibit the FAAH, the fatty acid amide hydrolase, and therefore it's almost like you're increasing the amount of cannabinoid in the synaptic cleft. So we'll come back to visit that point later. So where are these localized in the GI tract? And this has already been covered by my colleagues, the other speakers, and the cannabinoid receptors CB1 and CB2 are certainly present in the enteric nervous system, the blood vessels and the smooth muscle layers, and also in the GI epithelium. And it's not just the receptors, but also these enzymes have been localized. Enzymes for the synthesis and also the enzymes for the degradation or metabolism of the endocannabinoids. And in inflammation, there's increased expression in the epithelium as well as in the immunocytes in the lamina propria. How do they affect the functions and the activities within the GI tract? Well, they have effects on immune and inflammatory mechanisms, neuromuscular and sensory functions in the digestive organs. Let's look at that in a little bit more detail. Now, you've seen this slide earlier, and Keith Sharkey and John Wiley have this beautiful review in gastroenterology that illustrated that the effects of endocannabinoids mediated through CB1 and CB2 receptors ultimately result in reduced inflammation, reduced motility, and also changes in the permeability or the barrier function of the intestine. And this is another beautiful review published in our journal Gastroenterology on the effects of cannabis, and it summarizes many of these principles. The reduction in motility is shown by, for instance, a decrease in lower esophageal sphincter pressure, or which could result in more reflux disease. But on the other hand, reflux is related predominantly to transient lower esophageal sphincter relaxation. And so by decreasing these relaxation episodes, there should be a reduction in reflux of acid from the stomach into the esophagus. Now, we all know, and we're going to show you the data as well, that cannabinoids and cannabis in particular can decrease gastric emptying. But on the other hand, it improves its nausea, and we'll see the data from, for instance, the NIH Gastroparesis Consortium showing the use of cannabis by many patients with gastroparesis. There's decreased colonic motility, and I think, Keith, you showed these data as well, showing decreased motility and changes also in abdominal pain. And I'll show you data from a clinical trial from Bologna in Italy showing a potential beneficial effect with cannabinoid agents. There are also effects to reduce inflammation in the pancreas and also diverse effects in the liver that may actually have a role in fibrogenesis, which of course is very important because fibrogenesis is the precursor for the eventual development of cirrhosis. Potential utilization in human cannabinoid therapy. Let's review what we know very briefly about disorders of motility, pain, and functional disorders, as well as the randomized controlled trials. I want to remind you of this very important point, which is that cannabinoid receptors are not only expressed at diverse sites in the wall of the intestine or the GI tract, but also, I think very importantly, and this is where there may be a very significant potential effect, not only on the role of sensory pathways in the brain, but also an area which I believe is insufficiently emphasized, the role of the first order neuron that's going either from somatic nervous system or from the GI tract. This first order neuron is and particularly the dorsal root ganglion, which is the cell body of those first order neurons, is richly involving cannabinoid receptors. And I believe that this is probably a very important, as well as, you know, the effects on the spinal cord and the effect in the brain. At all levels, there are potential effects on sensory mechanisms that are important, for instance, in mediation of nausea, but also of pain. There have been some studies of the role of cannabinoid agents in chronic pain. So this is a study published several years ago that actually looked at 3 types of chronic pain: chronic abdominal pain, pain in chronic pancreatitis, and pain in the post-surgical patient. Chronic pain can sometimes develop, and unfortunately in these studies there was no significant difference between THC and placebo. But another study that came from the group in Bologna in Italy has shown that this substance, palmitoylethanolamide, together with polydatin, together have an effect on IBS, abdominal pain, and discomfort severity. So what is this compound? This is a compound that is structurally related to the endocannabinoid anandamide. And this is a rather complex figure where they plotted the information on pain sensation every 4 weeks. And what I want you to focus on is the fact that this combination is associated, the red lines, with a general trend for an improvement in pain over time compared to the effect of placebo, which basically doesn't change very much in this particular trial. So there is some evidence of reduction in abdominal pain severity in patients with irritable bowel syndrome. What about gastroparesis or stomach function? So there are studies that have shown that in healthy volunteers, tetrahydrocannabinol delays gastric emptying, and the non-selective agent dronabinol delays gastric emptying of solids. And I'll show you these particular data from the papers that have been published. And the NIH Gastroparesis Consortium has shown that 11.7% of patients who entered into this large multi-center study of gastroparesis, 11.7% are current cannabis users, and there is a perceived benefit for the severe nausea and abdominal pain. So let's look at the primary data themselves. So this is data from a colleague who unfortunately passed away only this year, Dr. Richard McCallum, and it showed that the THC was associated with a slowing of gastric emptying of solids from the stomach when traced over a period of 2 hours. We had done this study here at Mayo Clinic showing that dronabinol was associated with a retardation of gastric emptying of solids, and this was particularly evident in female volunteers rather than in male volunteers. Marijuana use is very common in patients with gastroparesis. So as I said, 11.7%, patients, very similar prevalence in idiopathic and in diabetic, as well as those who have delayed or normal gastric emptying. These are perhaps not really gastroparesis patients, but they have the symptoms of gastroparesis. They may be called functional dyspepsia in other settings. The patients using marijuana tend to be younger, current tobacco smokers, were less likely to be college graduates, or having incomes of greater than $50,000. And patients with severe nausea and abdominal pain were more likely to use marijuana and perceive it to be beneficial for their symptoms. Another prospective study looked at a cohort of patients, 24 patients with gastroparesis and refractory symptoms, and they looked at what is the benefit or lack of benefit of dronabinol, medical marijuana, or both. And they showed, as you can see here, a benefit after being treated with these cannabinoid agents compared to before. And this referred to the overall score, as well as in particular, this fullness and satiety score and the nausea and vomiting scores after treatment rather than before treatment. Now, this is not a randomized controlled trial, but it is a prospective cohort showing the beneficial effects of the composite overall score as well as the subscores. And particularly, I'd draw your attention to the fullness, satiety, and the nausea and vomiting scale. And there's a reason why I show that, because in our randomized controlled, placebo-controlled trial, we actually showed very similar effects. So this was a study that we conducted with the help of a grant from the NIH. And this was the time soon after Epidiolex or cannabidiol was approved for the treatment of a seizure disorder in children. And we were attracted to do this because this was a time where we knew there was reagent grade, as it were, cannabidiol that was approved by the FDA, and therefore we were quite confident about the standard and the consistency of the CBD that would be administered to these participants. And these were patients with idiopathic or diabetic gastroparesis. And we did a 4-week trial with a maximum dose of 20 milligrams per kilogram. So it was quite a high dose, highest dose that was permitted by the FDA. And we followed the dose escalation approach that was recommended, and we addressed daily symptoms using the American Neurogastroenterology Motility Society Gastroparesis Cardinal Symptom Index Daily Diary. So we assessed their gastric emptying at baseline. We also assessed the gastric emptying at the end of 4 weeks, and we assessed the other features that suggest postprandial symptoms, as I shall explain in the next few slides. So this was the trial that we performed, and this was the instrument, which is the FDA-approved Cardinal Symptom Index, originated from the American Neurogastroenterology Motility Society, but this is now in the FDA guidance for the treatment of gastroparesis. And the main points are to assess the patient's symptoms of nausea, vomiting, inability to finish a normal-sized meal, feeling full after the meal, bloating, upper abdominal discomfort or pain, and then the number of episodes of vomiting in the past 24 hours, and then a very useful overall assessment where the patient is asked, how severe are your gastroparesis symptoms today? And this is the adjectival scale that was used to assess the severity of symptoms. And the patients randomized to cannabidiol or placebo were basically comparable from all of the perspectives that we were able to look at, including these genetic variants for the gene for fatty acid amide hydrolase, as well as the cannabinoid 1 receptor, commonly found SNPs, single nucleotide polymorphisms. And so what you can see here is that we were very fortunate that randomized patients in the two groups turned out to be very, very comparable and well matched. So this is the main results that summarized on one slide. So I want to illustrate to you that the first data set refers to cannabidiol, the second data set to placebo. And this refers to a change in the patient response from baseline. So if you go below the 0 line, you have benefit compared to the baseline. And cannabidiol was associated with a significant reduction in the total gastroparesis cardinal symptom index compared to placebo. You can see that the median for placebo was virtually unchanged. And of course, there's going to be variance, as you see in any clinical trial. Some patients improve, some patients do not improve. The gastroparesis severity, which is a good summary indicator of the overall symptoms of the patient, was also significantly improved in the cannabidiol group compared to placebo. And then we looked at specific points with regard to the symptoms. The ability to finish a meal was improved. The sensation of fullness was somewhat improved numerically, but not statistically. The upper abdominal pain was somewhat improved as well. The frequency of vomiting in the past 24 hours, the number was statistically significantly improved, although relatively small numerical change. And there was no change that we observed in this trial for the symptom of nausea. So those were the symptoms that we collected with the FDA-approved gastroparesis cardinal symptom index. We also did some studies to look at what was the tolerance of food. And the way we do this is called the nutrient drink test. We almost use it as a way to assess how are the symptoms of the patients, what is the volume of Ensure, which is 1 kilocalorie per milliliter, that can be ingested until the patient says, yes, I'm comfortably full. That's an index of number 3 on the 0 to 5 scale. And then index number 5 says, I've got maximum tolerance. I cannot drink any more of this Ensure because I'm going to vomit or I'm going to have very, very severe symptoms. So here are the data. For that kilocalories to fullness, you can see that the patients given cannabidiol were able to tolerate at least 150 kilocalories more during this study compared to the placebo group. And the maximum tolerated volume was almost 350 kilocalories more with the cannabidiol compared to placebo. Again, consistent with what we had shown, which is that they're able to tolerate an ordinary sized meal much better compared to the placebo group. Now, as we also expected, cannabidiol did slow gastric emptying, and this has shown, the primary endpoint of the study was not significantly different, but we have to be totally honest. The amount of food retained in the stomach at 2 hours and at 4 hours were significantly greater. Sorry, the amount emptied was greater with placebo than with cannabidiol at 2 hours and at 4 hours, suggesting therefore that the cannabidiol did slow the gastric emptying. Now, we asked ourselves, if there was this slowing of gastric emptying in patients with gastroparesis, how might the cannabidiol have been improving the patient's symptoms? And of course, some of the comments we've heard from the other speakers have explained this very much more detailed than I can. But we saw from the publication in the literature that there were very important effects that could be reducing the symptoms, not only anxiety, but also pain sensations through effects on 5-HT1A, which we know is associated with symptoms related to the accommodation of the stomach, CYP-V1, which we know is associated with pain sensation. And I just want to remind you that even though cannabidiol is an antagonist of the CB1 and CB2 receptors, because it inhibits the fatty acid amide hydrolase, the endogenous anandamide is going to be increased. And we believe that this is how the one of the other effects of the cannabidiol to enhance the endogenous cannabinoids to reduce the symptoms in part due to the effects on other receptors, including dopamine, 5-HT1, and CYP-V1. We did do a very small pharmacogenetics analysis, and we did see that the variation in the fatty acid amide hydrolase gene was actually associated with the maximum tolerated volume, as you can see here. But this was really preliminary evidence that obviously needs to be confirmed in much larger studies than the one that we were able to do. Side effects, and this is interesting, and I keep asking myself whether I really know the mechanism, but it was interesting that about 50% of the patients receiving cannabidiol actually had diarrhea. And I'd be interested to hear what Keith Sharkey and the other colleagues on the panel would think about in terms of the reason why there was such an imbalance in the number of people receiving cannabidiol who developed the diarrhea. Perhaps from studies that we did before with dronabinol, there's an inhibition of contractility, maybe a reduction in tone, an increase in compliance of the colon, which reduces the resistance caused by the halstrations of the colon and therefore facilitates the passage of content through the colon, which manifests as diarrhea. But that's my speculation. I don't have evidence to confirm that I understand the mechanism, but I'd be interested in my colleagues' explanation or suggestion of mechanisms. So my conclusion, cannabinoid mechanisms modulate immune, inflammatory, neuromuscular, neurosensory functions of the digestive organs. In patients with gastroparesis, we know from the studies that have been done in patients that cannabis is used for symptom relief. We know that the cannabis, dronabinol, and cannabidiol all slow gastric emptying, and yet there is improvement in symptoms in these patients. And in our randomized controlled trial with cannabidiol, we showed symptom relief, total gastroparesis cardinal symptom index scores. The ability to finish a normal-sized meal was improved. The number of vomiting episodes was decreased. Overall symptom severity of gastroparesis was reduced. And objectively, we found improved tolerance of the liquid nutrient intake despite the slowing of gastric emptying. So thank you again for the invitation to participate, and I'd be very happy to participate in the discussion as well. I shall stop sharing, and thanks again.
Thank you very much, Dr. Camilleri. And I want to take this time to thank all of the presenters, including Dr. Mohan, who unfortunately could not make it, but I'm sure he will be watching this recording. We've seen that there is certainly a great deal of there's a big interaction that exists between the endocannabinoid system, the GI system, the human microbiome, the gut-brain connection, and we really appreciate those of you presenters tonight who help kind of continue to provide research and education around this topic, because I do believe that it's going to be increasingly important to individualized care. We are short on time tonight for a Q&A, so I think I'm just going to ask each one of the presenters to answer one question from one of the members in the audience, which I think will be very interesting to hear from all three of us, and that is, what research would we like to see that could further elucidate the relationship between cannabis, the gut-brain connection, and the role of the human microbiome? I will start by saying that I would love to see some research that looks at the individual microbiome profiles and to see what the actual serum levels of circulating plant-based cannabinoids are in the context of individuals with varying microbiomes. So, for example, we know that the microbiome, certain bacteria can metabolize cannabis. It can cause cannabinoids, which were inactive, to potentially be reactivated and enter the circulation. And as a clinician, I would love to know if somebody's human microbiome, if the composition of it would need to be considered when utilizing cannabis. Dr. Sharkey, what's a particular area of interest that you would love to see pursued further?
Yeah, it's a great question. It's there's a lot of areas that we really don't understand at all, and I think this comes down to understanding how cannabis in a real world setting, particularly as we've moved into this era of high potency cannabis that's now the major form that's available in all states that have legalized it, the whole of Canada, for example, and other countries. This has changed enormously how we view it. And most of the early work, a lot of the stuff I presented, for example, this is all on old stuff that isn't really what people are taking anymore. And I would love to understand both the impact of real world cannabis consumption on the microbiota in a naive user on a particular diet, as well as on a user who's now become tolerant to cannabis, because that's what will happen, and to see how their gut microbiota is altered or not altered, and then how that changes on an individual basis over time, to really understand how these things are interacting with one another. We don't have any sort of data of that nature.
Yeah, no, absolutely. I agree. And I look forward to this. I know it's a very complex field, and certainly with the advent of, AI and quantum computing and things of such, hopefully we'll be able to get to some of these answers quicker than we could have 50 years ago. Dr. Camilleri, as it relates to the endocannabinoid system in cannabis, what are some of your hopes that we'll discover in the future here?
Well, certainly, I don't want to belittle the importance of the endogenous and the cannabinoid system, but one of the thoughts I've had as I've studied this further is, I try to indicate the importance of that dorsal root ganglion cell and the role that this might have not only in somatic pain but also in visceral pain. And unfortunately, the biggest unmet need we have in irritable bowel syndrome and the patients that we see, for instance, with inflammatory bowel disease, where the inflammation has been treated, but the patient still has pain and tenesmus and other symptoms like that. So is there a way in which we can modulate the function of that dorsal root ganglion cell, which is taking, that first message of pain from the gut or from the somatic nervous system back to the spinal cord and up to the brain? Isn't that an opportunity for us to be able to modify the sensation, somatic or visceral, as afferents, in order to reduce pain, which in my mind is still the major unmet clinical need that we have in the area of functional gastrointestinal disorders, or what are currently called the disorders of gut-brain interaction. So that was what I'm hoping, that there will be a reintroduction of interest in trying to modulate the visceral afferent system in particular.
Fantastic. Thank you very much. All right. Well, once again, I want to take this time to thank you both, Dr. Keith Sharkey, Dr. Michael Camilleri, and Dr. Mahesh Mohan, for presenting tonight. I know I personally learned a lot, and I really appreciate you helping our organization and everybody on this talk tonight to kind of help us think about this, as it relates to caring for, our patients, and things that we'll probably need to consider and keep our eye out here in the future of cannabis care. And having said that, I want to thank everybody who was able to tune in tonight. And again, this will be syndicated on the website for those of you who couldn't make it. From the Association of Cannabinoid Specialists, again, I want to thank everybody, and hope you all enjoyed the presentation and have a wonderful rest of your evening.